Cancer Research UK Cambridge Centre podcast. In this Integrated Cancer Medicine Research in Focus series, I talk to various ICM members about their research and how it is supported by the vision of the Mark Foundation Institute for Integrated Cancer Medicine. MFICM research uses cutting-edge analytics to maximise the use of diverse high-volume datasets and by capturing cancer heterogeneity in time and space in patients receiving active treatment. Integrated Cancer Medicine aims to transform the way the world treats cancer by affecting patients along their treatment pathway and ultimately accelerate cures. Today I have with me Professor Grant Stewart and Professor Ava Sala, and we are going to talk about the WIRE clinical trial. Professor Stewart is Professor of Surgical Oncology at the University of Cambridge, co-leader of the Integrated Cancer Medicine Programme and Chief Investigator for the WIRE trial. Professor Sala is Professor of Oncological Imaging at the University of Cambridge, co-leader of the CRUK Cambridge Centre's Advanced Cancer Imaging Programme and co-leader of the Integrated Cancer Medicine Programme. If we could just start with Grant, if you could just give me a bit of a background on what disease the WIRE trial focuses on, please.
Yeah, thanks, Ellie, and thanks for inviting us along to this podcast. I'm a kidney cancer surgeon, and all of my research focuses around kidney cancer pretty much. And I'm interested in the disease because it's the seventh commonest cancer in the UK. It's the most lethal of all of the cancers that I, as a urologist, encounter. So it's more lethal than the much more common prostate cancer and also bladder cancer. And it's a cancer that presents very quietly. So like ovarian cancer, like pancreatic cancer, the majority of our patients have few symptoms often, and they often don't have any relevant symptoms. So they don't have the sort of red flag symptoms. So it's a cancer type that really needs a lot more research, because if we can identify the disease early, we can cure the disease with surgery. But equally, if we identify the cancer later on, when the tumor is larger, we need some better ways of ensuring that surgery can be effective as a curative treatment. And that will need other treatments in addition to the surgery.
And is this the focus of the WIRE trial, these other treatments?
Yeah, that's right. So that falls into our interest of giving drug treatments in and around the time of surgery. So you can either do that before the operation or after the operation in what's called adjuvant treatment. So the WIRE trial is a study where we give drugs to patients who are eligible and interested in participating before surgery. Before surgery, you can either give a short course of drug medication, and that's what we're doing in WIRE, or you can give a longer course, which is called neoadjuvant treatment. So WIRE is this short course of drug treatment in this so-called window of opportunity between diagnosing the cancer and being able to get our patient through to the operating theatre and undertake the surgery for them. And
a little bit about the trial. How is the trial funded?
Yeah, so this is fairly novel, I think, in the grand scheme of clinical trials, because it's really funded from three different sources, all working in collaboration. So it's funded through our CRUK Cambridge Centre funding. It's funded with our partners AstraZeneca, who the initial drugs in the trial are all supplied by AstraZeneca. And it's also funded by the Mark Foundation. And as you mentioned, we're part of the Mark Foundation Institute for Integrated Cancer Medicine.
And when did it open?
We had our first patient first visit in September of 2020. We opened just a month or so beforehand. Obviously, we had a little bit of hold up last year because of wave one of the coronavirus pandemic.
Sure. And so how many patients have you got currently recruited, and how many do you aim to recruit in the future?
Yeah, so we've been absolutely thrilled by how well this has been received by patients. So we have had 7 patients complete the trial already. And for this type of study, that in my experience, is pretty remarkable, actually. And that's considering the issues we've had in our hospital, as many hospitals have, around coronavirus. That's been very good. And actually, that's not only been great interest from the patients, but it's also been because we've been able to coordinate with all of the colleagues required to do this quite intricate study. And Evis has made sure that we've got all of the radiology that we need, which is multifaceted. It's getting a biopsy promptly. The study involves MRI scans before starting drug and at the end of the drug treatment. And she and colleagues have ensured that we've got those in a very timely fashion and that we're able to do the image processing, which she'll speak about at some point.
So perhaps we can talk about the treatments now. What is the current gap in the research, and how are you plugging that gap with these novel methods and innovations?
At the moment, if somebody is identified to have a mass in the kidney, the majority of people end up having surgery, and 50% of all patients with kidney cancer will eventually die of the disease. So although the surgery is initially beneficial, many of those patients will have a recurrence later on, unfortunately, and there's very little we can do. There are no proven treatments to prevent that. There are no proven adjuvant treatments given after surgery, although there are clinical trials going on in that area. So we're giving the pre-surgical treatment, and we hope that this will benefit people in terms of a better survival from kidney cancer in the long run. So the treatments that we're giving in Wire is a platform for us to offer our patients new innovative drug treatments that haven't been used before in kidney cancer. And we are offering them treatments that we've got a good biological rationale from Cambridge and AstraZeneca science that they will be beneficial. The main drug type that we want to offer to patients in Wire are the DNA damage response inhibitors. And that's been a really interesting area to explore with AstraZeneca, because these are not drugs that you would typically think would be active in renal cancer, because renal cancer patients don't tend to have a very high DNA damage response genetically, unlike, say, breast cancer or prostate cancer. However, there's a lot of biology that we've recently discovered that suggests that, in fact, these cancers are going to be susceptible to those drugs. So, for example, the key genetic mutation in renal cancer, which is the VHL gene, the von Hippel-Lindau gene, occurs in virtually all people who get kidney cancer. It turns out that actually provides susceptibility to having DNA damage response inhibitors be effective. So we think that along with some other mutational information, and also the fact that if you have this initial hit of hypoxia, low oxygen conditions in a tumor, which we know occur in renal cancer in virtually all cases, that then those tumors again will be susceptible to DNA damage response inhibition. So it's this biology that hadn't previously been highlighted that has provided the rationale for us offering DNA damage response inhibitors, so olaparib essentially, from the AZ portfolio in Wire. So we're using olaparib, but we're also using it in combination with other medications that are known to work well in renal cancer, and we also think will further potentiate the effect of the olaparib. So those drugs are in two classes. One is a drug called cediranib, which is an anti-angiogenesis drug and reduces the blood flow to the very small blood vessels to the cancer. And secondly, the immuno-oncology drugs, the T-cell checkpoint inhibitors, which are very effective in advanced kidney cancer. Again, there's biology to suggest that in combination with a DNA damage response inhibitor, that the effect of those two drugs together will be synergistic. So they'll work better than either one on their own. So that was the rationale for this platform.
And how does your work fit in with this, Evis? If I may throw it open to you, can you describe the techniques that you've been using?
So there are several techniques that we're actually going to apply for the first time in these trials. We're all very excited about it. Some of the development work was done in ovarian cancer, but this is the first time we're actually applying the techniques and evaluating them in the context of the clinical trials. So what we're doing in the WIRE trials, actually, for the first time, implementing some of the computational methods, but also imaging tools that we have developed as part of the Advanced Cancer Imaging Program, but also Integrative Cancer Medicine Program. So a patient will have a biopsy prior to starting the treatment of the trial. Traditionally, the biopsy is guided by imaging to ensure that the biopsy is safe and it actually samples any part of the tumor. With the WIRE trial, for the first time ever, we're actually using a new method that not only samples the tumor and is safe, but also samples different parts of the tumor. What I mean different, mean that they look different on imaging, or so-called tumor habitats, because they have a different combination of blood flow, of tissue or tumor permeability, of the cell density. So we'll be able to actually have a more in-depth knowledge of what's going on in the tumor, but also ultimately when this patient comes at surgery after they've completed the course of treatment, we'll be able to track those changes that happen in a tumor. And because we'll have sampling at both biopsy and surgery, we can also look at the tissue, what has happened. That in the future, I'd hope, will help us establish the virtual biopsies. So we really don't have to biopsy patients multiple times in the trial, but just by looking at imaging, we can actually tell if the drug is working or not. You know, it's not current immediate, but this is what we'll aim in the future. Obviously, those patients will undergo MRI, which will have some anatomical images, but also will have some more advanced MRI, like diffusion MRI, which tell us a bit more about the physiology of the tumor. So we've developed computational methods that can extract quantitative data from all these MRI sequences, and they can combine them together to generate those imaging habitats, which are essentially clusters of different combination of those parameters. Then the images with the MRI, and then those habitats which are overlaid on the MRI are sent to the ultrasound machine, which is obviously in the clinical department. So when patient arrives for the biopsy, those habitats are loaded into the machine. And then as the biopsy is taken, the radiologist will sample those areas guided by the imaging habitat. So you overlay the MRI with the imaging habitat with the ultrasound so that you can really sample them. So these new tools and computational methods are developed and then applied before the treatment is started, just prior to surgery. In order to track those changes in imaging, we've actually developed another method to sample the tissue. So we develop 3D printed molds that are not only patient-specific, but they're also tumor and kidney specific. So the whole kidney with the tumor, as it comes out, is put on these specific molds, which are really designed based on the MRI. So now, as the pathologist is actually sampling the tumor, they can sample accurately the areas that we are following up on imaging, various parts of the tumor. So you can not only spatially trace or track the changes treatment, but also temporally. So we can eventually understand in a non-invasive way what is happening to the tumor.
Yeah, and I think it's important to say that we've done these individual elements before in kidney cancer and ovarian cancer. I think WIRE is a great example of putting our very best science into a clinical trial to see what clinical benefit we can give to our patients and better understanding of the tumours and such like. And so this is the first time, I think, that we've put all of these elements together in a clinical trial. It's the first time globally that's been done.
Exactly. So the reason we're super excited, actually, we are applying all this in the context of a clinical trial. So we've now brought together the computational part, so the quantitative extraction of imaging data, the novel AI algorithms to actually generate those habitats, and the tools to actually sample the tissue precisely. Another a new element to the trial is the hyperpolarized MRI, and that is adding another layer of understanding of what's going on in the tumor, because this is a very quick imaging of the tumor metabolism. So what we want to do is quickly see what happens to the metabolism in vivo, so with the tumor in the patient, before and then obviously following treatment. And this is really work that's spearheaded by my colleague, Professor Ferdia Gallagher, has been done in a few cases before in breast cancer, mainly in the partner trial. But this is really the first time done in a proper way to combine the anatomical, the physiological, the metabolic imaging, the computational part of it on top to insert those tools to actually sample the tissues.
So this is a significant change in the patient pathway. And how do you think it will impact the patient treatment outcomes?
So you're right, it is quite a significant change, but it's interestingly, it's almost what patients expect to happen. So they're aware of friends and family who've been treated for other cancers that have had biopsies and repeat imaging and have had drug treatments before surgery. So in many ways, this is not something, although it's different for us as clinicians, because this isn't the normal NHS pathway, if you like, our patients are unfazed by it. It's important to say that the primary endpoint of WIRE is not patient survival, because we're giving a very short treatment of drugs in that window of time, which is 1 month, between when I see a patient in clinic and tell them what's happening, there's a cancer, it needs to be removed by surgery, and then 1 month later them having that operation. And that month is almost always the case, because that's how long that's our capacity in the hospital. Those are our NHS cancer targets. And so we have the exact number of surgeons and operating lists to deliver that. So they get a short period of drug. We can't look at survival in the relatively short follow-up time frame that we have. That's for a future study. But what we're assessing, once again, we're going back to radiology, where we know we've got great surrogate endpoints. And so we're measuring radiological characteristics that look actually at the very small blood vessels once again, and how leaky they are. And we expect that should change over quite a short period of drug treatment based on our previous studies in previous clinical trials, like the NEOSUN study, which was undertaken by Professor Tim Eisen, and Evis and Ferdia Gallagher were involved in that. So, we're putting all of these tools into use once again.
And do you think that this new paradigm will be accepted into standard of care eventually?
There is more effort from the computational team and preparation done before that, but actually, in terms of patient experience, it will only add 10 minutes in the procedure room. The other thing we've done with the 3D molds, we've made it so it doesn't disrupt the process of the pathologist processing the tissue. It adds an extra layer of providing a mold, like a little plastic box in a way. Other than that, the tissue is processed exactly the same way. So the information that is needed for staging purposes doesn't have to put a bigger extra effort to actually process the samples. This is really important because there's a patient acceptance, but also it's a workflow acceptance.
So actually, just to build on that, the acceptability seems to be very, very high. Fortunately, there haven't been too many side effects from the medication either, which is good. And that shows the skill of our oncologists in making sure that the dosing is right and that the side effects, if there are any, are managed promptly and well. So I think it will be very well received and accepted by patients if this becomes standard of care in the future. I think Evis's point is absolutely critical, that clearly in this study, we're biopsying more people than we do in standard of care, let's say. That's one example. We're imaging at an extra time point to what we would normally do. And there's a little bit more work from the pathologist. And that's fine in a centre like ours, where we're kind of well set up to do this. We've got a very strong research profile. We've done a lot of these things piecemeal before, and now we're putting them all together. What's going to be really interesting, though, is to see how the processes work in the other 2 centres that we're hoping to open very shortly, and they're Glasgow and the Beatson Institute there, and Oxford, where we have very strong links with, in particular, with the surgical oncologists. So there we will see which parts of the pipeline could potentially cause problems. The other exciting thing about this multi-center nature of the study is, once again, coming back to the radiology, I think, and Evis will correct me if I'm wrong, but I think this is the first time ever that we've done a multi-center study with hyperpolarized MRI, which is quite exciting.
Absolutely, this is absolutely correct, and it's exciting. First because we can apply technology in the 2 centers, and then also I think in order to make these technologies viable, to actually make use of them in routine clinical practice, we have to go beyond a single center.
Can I ask both of you in turn how this fits in with your broader research? Perhaps, Grant, if I could start with you.
The mission, if you like, for my research program is optimizing the management of initially localized kidney cancer patients. And I term it like that because unfortunately, despite our best efforts with surgery, and that's the workhorse treatment of patients with localized kidney cancer, there's no radiotherapy or systemic therapy as we've been discussing. The facts are that despite my best efforts, about 30% of my patients will go on and have a recurrence later on. I've got two main components to my research. The first is around early detection and screening for kidney cancer, and we have a significant amount of work going on there, moving through to this sort of peri-surgical systemic therapy. So as well as the WIRE trial, we've just completed a study called NAXIVAR, which is a clinical trial, once again giving drugs to patients before surgery. This was a multi-center UK study that I led from Cambridge, where we gave 8 weeks of a drug called axitinib to these patients and reduced the extent of locally advanced kidney cancer. And that's given some really interesting results that we're about to present in an oral presentation at ASCO GU. So, we've got quite a good track record now doing these pre-surgical drug trials. And then building on that, it fits in beautifully with the adjuvant studies that I'm involved with, the main one being the RAMPART clinical trial, which is an exciting statistical design called a multi-arm, multi-stage clinical trial, where we can change arms of the study as the study goes along. And it's randomizing standard follow-up after surgery to immunotherapy, either one immunotherapy or doublet immunotherapy. I should just touch on one aspect related to that, which is the statistical design. So I've described that has quite a funky statistical design, but WIRE also does. One of the very cool elements is that we have James Wason, who's Professor of Biostatistics at Newcastle and still has links with the MRC Statistics Unit in Cambridge, who has designed the statistical elements. And it's a Bayesian adaptive design. And what that means essentially is that we give as few patients as possible the drug treatment in each arm until we've proven that it either works or doesn't work. So we have various stages of recruitment. So we've just met the first stage of recruitment in the first arm, which was 6 patients. We then analyze those patients and see whether we need to carry on and recruit more patients in that first drug arm, cediranib, or in fact, we can formally move on to the second arm, cediranib plus olaparib. We're sort of doing it in real time, which is almost unheard of in clinical trials for you to get endpoint data within a few weeks, but we're doing that right now. But what's very cool is that we continue to recruit by opening the second arm. So our 7th patient went into the cediranib olaparib arm. If we need to reopen the cediranib arm, we can do that. We just pause that second arm and come back to it once we get to that point. So again, fits in really well with the broad mission of my research, and once again brings in these very exciting bits of research that we've done over the last few years: imaging, statistics, and the translational work. We haven't talked about that, but ctDNA, metabolomics, working with the Sanger and Tom Mitchell, who's an expert in single cell transcriptomics and genomics, and putting all of that together in a clinical trial is extremely exciting.
It's a real example of integrating so many different streams of data in order to form conclusions. And Ivis, if I could just throw that question out to you, how does this fit in with your broad research?
At the end, it's sort of an example of how would I want imaging to actually be playing a role in medicine, especially in the context of data integration. I always had the vision of you can extract more from images that you can see. And so far, if it's any stream that we haven't actually used properly quantitatively, it's been imaging, because we tend to be qualitative and, describe what we see, but actually all that quantitative information is not used. So that's why as I started here 3 years ago, I created a quantitative and radiogenomics imaging group, which really has developed this computational methods, obviously in strong collaboration with the Cancer Institute, with the Department of Applied Mathematics and Theoretical Physics, with engineering. So it's been a huge collaboration of various people, and of course with the great support from Grant in case of kidney cancer. And now we have a platform that we can do this in a way that it can be really integrated in a clinical trial and in a clinical practice as we move along. I think also it fits because I really want to sit always on the border. So what I do, it has to be clinically translatable. But I think I want to see the imaging making a difference, or my research making a difference to the patients now and very near future.
My last question really to both of you is, where do you see integrated cancer medicine taking us in the next 5 to 10 years? Evis, perhaps we can start with you this time.
What I would like to see, and I'm sure we are going to achieve that, is that what we develop and will continue to develop in terms of methods, tools, new tracers, new probes, they can actually be implemented into clinical practice. Yes, of course, some of them will be through clinical trials to start with and then clinical practice, but I want very much to see them as part of this routine trial or routine clinical patient work. And I think what is even more exciting that we're going to have a cancer research hospital in the next 5 years. So even physically, that will happen under the same umbrella. So I see everything coming together. And in 5 years' time, we're going to have this really novel clinical trials in terms of design and the compounds used in the clinical trial, but also novel tools, novel imaging, novel ways to look at the data, novel ways to predict the treatment response, novel ways to predict the outcome. And then ultimately, novel ways to actually visualize all of that in our multidisciplinary meetings. So I think the future is fabulous.
Thanks. And if I could ask the same question to Grant.
Yeah, so I agree with everything Evis has said, and she's basically taken the words out of my mouth for a lot of this. A couple of extra points. One is we've in Cambridge and actually now in many other trusts around the UK, we have fantastic electronic patient records, and we are not able to take advantage of those systems currently. So both in clinical trials and also hopefully very soon in routine clinical practice, integrated cancer medicine approaches, as is being spearheaded from Cambridge and with the Mark Foundation support as well, is going to allow us to just absolutely make the most of that information. So, an example would be, I can look at a scan with Evis and we can choose over what we think of the patient's tumour, but understanding the physiology of the patient and how their disease and the patient's performance fit together in what would be the best treatment is an intuitive thing that I do with my radiology and pathology colleagues, for example. That's okay, but in borderline cases, it can be very, very challenging. And so that's where integrating data and giving us new tools to be able to improve on what we do currently is going to be supremely useful. And then I think the other thing that really is the strategy we've taken with Wire is integrated cancer medicine is a key way that we can integrate the most exciting of our basic and translational scientific discoveries eventually into routine clinical practice, because we actually have to find a way of making this work, of getting those discoveries from a bespoke basic scientist benchtop into a routine clinically facing laboratory and to the patient. That's just one example. So I think that this is the vehicle that we can use to get these things into clinical practice.
That's excellent. Thank you both very much for joining me today on this podcast. It's been so interesting to hear all of your ideas and research, and particularly about the Wire trial. Thank you.
Pleasure. Thank you.
If you want to find out more about the work of the Mark Foundation Institute for Integrated Cancer Medicine, please visit our website at www.integratedcancermedicine.org, where you can find details of the ICM vision, all the current research, clinical trials, resources, publications, and team information. You can keep up to date with our latest news and events, and you can also sign up for our newsletter. If you would like more information about the work of the CRUK Cambridge Centre, please go to www.crukcambridgecentre.org.uk, or you can connect with us on Twitter using our handle @CRUKCamCentre. Thanks for listening, and do join us again soon.